Hello students, today we will discuss about the protein and its structural organization. Proteins are the most abundant organic molecules of the living system. They constitute about 50% of the cellular dry weight and the fundamental basis of structure and function of life. The proteins are nitrogenous macromolecules that are composed of many amino acids. Amino acids are a group of organic compounds containing two functional groups, that is amino and carboxyl. The amino group is basic while the carboxyl group is acidic in nature. There are about 300 amino acids occurs in nature. Only 20 of them occurs in proteins. Now discuss the structure of amino acid. Each amino acid have four different groups attached to a alpha carbon, which is a C atom next to COOH. These four groups are amino group, COOH group, hydrogen atom, and side chain, which represented by R. Acid has four groups. Number one is non-polar, which is hydrophobic. Number two is polar, which is hydrophilic. And number three is acidic, which is negatively charged, and basic, which is positively charged. There are four levels of protein structure. Number one, primary structure. Number two, secondary structure. Number three is tertiary structure, and number four is quaternary structure. Summary of the four levels of protein structure using hemoglobin as an example. Now, the first structure organization of protein is primary structure. Primary structure is a linear arrangement of amino acids present in the polypeptide chain. It starts amino N terminal and ends with carboxyl C terminal ends. Peptide bond is rigid and planar. All peptide bonds in protein occurs in trans configuration. The rotation is permitted about the bond between nitrogen and alpha carbon atoms and between the alpha carbon and carboxyl carbon atoms. Forces are involved in stabilizing primary structure include covalent bonds, peptide bonds, and disulfide bonds. Now the second is secondary structure. Secondary structure, is a regular folding of primary structure is called secondary structure. Folding and hydrogen bonding between neighboring amino acids results to form rigid and tubular structure called helix. Pauling and Corey studied the secondary structures and proposed two conformations, number one alpha helix and beta sheets. The forces are involved in stabilizing secondary structure include hydrogen bonds, peptide bonds, and disulfide bonds. Basically, alpha helix is a coiled structure. Peptide bond would form a right-handed helical structure by simple twist about the alpha carbon to nitrogen and the alpha carbon to carboxyl carbon bonds. It can be right-handed or left-handed and stabilized by hydrogen bonded to the NH and CO group of main chain. In this picture, each green circle represents an amino acid. Chains of amino acids form a spiral or helix structure. Now the second is beta pleated sheets. In this picture, each red circle represents an amino acid. Chains of amino acid bonds to each other. Beta pleated sheets form when two or more polypeptide line up side by side. Individual polypeptide beta strand. Each beta strand is fully extended and they are stabilized by hydrogen bond between NH and carboxyl groups of adjacent chains. Third structure is tertiary structure. The tertiary structure of protein is the overall three-dimensional shape that arises from all of the secondary structures of its polypeptide chain. They do not occur randomly, and the forces involved in stabilizing tertiary structure include electrostatic forces, van der Waals, hydrogen bond, and disulfide bonds. For example, myoglobin. Myoglobin is a relatively small, compact macromolecule, oxygen binding hemiprotein, and found in muscle cells. Molecules contain single polypeptide chain on one of 153 amino acid residues. And the last structure is quaternary structure. The quaternary protein structure involves the clustering of several individual peptides or protein chains into a final specific shape. A variety of bonding interactions, including the hydrogen bonding, salt bridge, and disulfide bonds, hold the various chains into a particular geometry. Two kinds of quaternary structures, both are multi-subunit proteins. Number one, homodimer, and number two is heterodimer. The interaction with multi-subunits are same as that found in tertiary and secondary structures. Forces are involved like disulfide, hydrogen, hydrophobic, and ionic bond. Example is hemoglobin.